Hey everybody, Mr. Bress here, and I just want to give a recap of what we talked about in class today, where we introduced codominance. And like I said in class, this is one of my favorite topics in genetics because it brings some things we've all heard about in the world and brings it to, sorry, I got to get my view right here, and brings it, oh, what am I doing here? And brings it to how we can solve problems using it. So it takes something that you maybe heard about, and we learn a little bit about the basics of it, and then we learn how we can use that to solve some genetic problems. So before we get into that, I want to recap just a couple of the other versions of genetics that we had learned prior to codominance. That's the complete dominance and incomplete dominance. And so let's head over. I'm going to share my screen here. If this is working correctly. Let's see how this is going here. There we are. All right, now I'm feeling like everything's working. So let's head over to Notability and just draw out kind of what we talked about in class today. So I'm going to just start a new note. And when we started this unit kind of getting an overview of genetics. And that overview allowed us to see that we get half of our chromosomes from our father and half from our mother. And each one of those chromosomes carries about 1,000 genes per chromosome. And at each spot, you get one version of a gene from your father and one version of a gene from your mother. And in our world, we're just going to pretend for now that there's only two different versions of the gene. And then once we understand the real world implications as you move on in your scientific careers, you'll understand how to account for those. But here we're just talking about two versions of a gene. And the one we started with, we didn't call it this at the time, but is known as complete dominance. And incomplete, not incomplete, in the version of complete dominance, we talked about two different genes. And the one we started with was eye color. And we used a capital B to indicate brown and a lowercase to indicate blue. And in this scenario, we said, well, we kind of get it. If I get a version of the brown gene from my dad and a version of the brown gene from my mom, my eyes are going to be brown. That seems relatively straightforward. And it was not too hard for me to imagine that if I got the blue gene from my dad and the blue version of the gene from my mom, that my eyes would be blue. Again, not too hard for me to imagine that, just starting in genetics. The question then became, what about the heterozygote version? This is the heterozygous, hetero meaning different. What about the heterozygous version? Is one eye brown, the other blue? We've talked about this a few times. No, in the case of complete dominance, it only takes one version of the dominant gene and you have that complete phenotype. And so in this case, the version would be brown. So that was a pretty standard version that we talked about in complete dominance. Then we moved on to the scenario where there might be incomplete dominance. So incomplete dominance. And there's not a ton of examples of this in high school genetics or introductory genetics. I think there's many more in the real world, but the types of problems you see in high school genetics are a little bit limited. And typically the one that comes out the most is flowers, these snapdragon flowers. And in the snapdragon flower, there is a red flower, and that's signified by two versions of the red gene. And there is a, oops, I don't want fully white. That will be a little difficult to see. There is a white version, of the flower with two versions of the white allele, or two versions of the white gene. And then there is the heterozygous. You get one copy of the red gene and one copy of the white gene. And what does this become? Well, this we learned, the flower is pink. And we talked about how early geneticists, if they crossed a red flower with a white flower, all of the flowers, all of the seeds that came from that cross produced pink flowers. And we thought, oh, that seems pretty obvious, right? And then the early geneticists we talked about crossed, and this is cross-pollinated a pink flower with another pink flower. And they sort of expected, yeah, this will be easy. All the flowers, all the seeds for this, they're going to produce pink flowers, right? Well, we know now that that's not what happened. It was very surprising that it produced red, white, and pink flowers. And we recognize that this was because these ones were heterozygous. And in the heterozygous cross, if you cross RR crossed with RR, remember that these ones here are pink flowers, right? And you make that cross there, you get this combination here where you get the red, the pink, and the white. And so once we understood the genetics that dictates the flower color, it became a lot more obvious that that would be the situation that would happen. And so that is known as incomplete dominance. So the version we're going to learn about today is called codominance. Codominance. So here, codominance. In codominance has a couple really interesting applications, and one of those is blood type. So blood type, I'm going to write the blood type here. I'm going to write the phenotype here, the antibodies here, which will become clear in a second, and then finally the genotype at the end. So blood types come in 4 basic versions. So you can be A, you can be B, you can be AB, or O. And what does blood type mean? Well, it's the type of protein that's attached to our red blood cells. And so we could get pretty deep into this, and we got a little deeper into class, but red blood cells are kind of these disc-shaped cells that I'm sure if you looked around the internet, you're going to find much better versions of a red blood cell than what I'm drawing here. And red blood cells are important for a number of reasons, but probably most critically is they carry oxygen in our blood. And they kind of have this round disc shape, unless, of course, if you have sickle cell anemia, they become sharper and more of these crescent moon shape, which can be problematic for clotting in your blood. But let's focus on the healthy red blood cells here. And so what does the different types mean? Well, the type means, like I just said, is the type of protein the type of protein that's on the surface of the red blood cell. And this is a whole other area of study of what the surface proteins do on red blood cells, but they're involved in recognizing other cells in our body and responding to environmental cues. So they have a bunch of different features, but they kind of are these proteins that stick out of our cells. And so here is the A type of protein. And there is a B type of protein. And this is B. And in blood type AB, you express both of the types of proteins. Your cells express both of the proteins. And in type O, you have these incomplete proteins. They don't make the full protein, so they don't actually stick out of the cell. And what we talked about in class, and I don't want to get too much involved in here, is your blood has antibodies. Your immune system circulates these antibodies that go around and look for cells that should not be in our body. Okay, and we produce thousands of different versions of these antibodies, and they float around and they bind to things specifically that are not supposed to be in our bodies. And it's a fascinating area of study, the immune biology. But I'll just say this, that one thing we don't want to have happen is we don't want to have antibodies that recognize our own cells. So if an antibody binds to a cell, it sends out a signal that says this cell is not supposed to be here, and we mount an immune response to it. It's in fact really relevant right now when we're talking about vaccines and virus and responding immune there. So we talked a little bit about that in class, but you just don't want to have antibodies circulating that recognize your own proteins, because that leads to an autoimmune disease. Like type 1 diabetes is associated with autoimmune disease. Alopecia, if you lose your hair, that's associated with it. That's an autoimmune disease. And rheumatoid arthritis is another autoimmune disease. So you don't want to have those antibodies. So the possible antibodies that you could have, you have many, but let's just focus on the ones that could be recognizing our own red blood cells. So you might have, and I'm drawing this as a triangle as if it would fit into the A protein on the red blood cell. So you could have the A antibody, or you could have the B antibody. Okay, so we'll be more specific here. So these antibodies, if this antibody would kind of fit into here, and once it locks in there, it changes its shape and it sort of sends a chemical signal that says, hey, this is not supposed to be here, and the immune response is mounted. Some of this is a bit of an oversimplification, but immune system is a complicated process. So what we don't want to have is, if we are blood type A, our body does not produce the A antibody. And that's a whole nother reason of how that happens, but we don't produce that antibody. If we did, we would recognize our own red blood cells, we would kill our own red blood cells, and we wouldn't last very long. The same thing is if we're type B, we do not want to produce the B antibodies. And if we're AB, we don't produce either of the antibodies, because that would be problematic. They would bind and kill and lead to the destruction of our own red blood cells. In type O, we actually produce both of the antibodies because there's no reason not to produce those. And this becomes important when you're talking about blood transfusion or transplants when you're bringing in new organs and a different blood type and mounting a response against the organ transplant or the blood transfusion. So AB, in the case of AB, you don't produce either of the antibodies. So you can get blood, you can get the red blood cells from any other type of blood, and you don't produce an immune response against it. Type O, on the other hand, you produce both the antibodies. So any other blood that you might get in a transfusion, you're going to react to that because you produce both of the antibodies. The nice thing about O is, though, is your red blood cells, if you're type O, don't have the protein. So there's nothing for the antibodies to recognize. So that's why O is known as the universal donor. AB, on the other hand, is the universal acceptor. You can get blood from anyone. Otherwise, you want to just get a transfusion from someone that has your blood type. And so how do we sort of indicate this? How do we code for this, right? Because scientists aren't going to draw this every single time, and we're not going to put those into a Punnett square or a cross. So we have a little code for it. And for A, there's two ways to be blood type A. You could have gotten the A version from both your mom and your dad. And that's how we write this with this I and then the capital A. And this I here, this stands for immunoglobulin. And that's the name of the protein that's produced. So the I comes from immunoglobulin. So the two ways you could be type A is you could have gotten the A version from both parents, or you could have gotten A from one parent and the non-functional version, that's the one that comes in the O type, the non-functional version from one of your parents. If you get one non-functional and one A, every cell is you're still gonna be showing that A protein. The same goes for B. You could have gotten B from both parents, or B from one parent and the non-functional version from another parent. AB, there's only one way to get that, is you got A from one parent and B from the other. And O is expressed with you got the non-functional protein from one parent and the non-functional protein from another parent. So you could have problems where it says, what are the possible genotypes of an individual? Let's say it's the mom who is type AB, has a child with a father who is type O. What would be the possible genotypes of this offspring? Okay, so we do the Punnett square the same way that we had done before. And then we match up in the boxes, this, here, and here. And we see that there is a 50% chance, 2 out of 4, or 50% chance of having a child that is type A, and 2 out of 4, a 50% chance of having a child who is type B. Okay, and we talked about then that knowing this, actually this was used in forensic science early on, in sort of some early types of paternity tests. So if you had a mother, if we knew the mother was type AB and crossed with some father, and the child was also type AB. Let's say the baby is type AB. And the question is, could this individual have been the father? Well, if the baby is AB and the father is type O here, there's no way, there's zero chance that the baby here could be type AB. And so you would know that this individual here was not the father of this child right here, because there's no way you could produce that blood type. So you could go through some more complex problems, and we do some of that in the practice. So this is just a brief introduction to codominance for the blood type. Oh, I should say there's one other version of this, and it comes up with these checkered chickens and sometimes tabby cats. So, and I think I'm getting this right, but in a tabby cat, let's do this. So big T is tan. And what we're going to say is we're going to call this T prime. T prime is black fur. This is in cats. And in this case of incomplete dominance, if you have T and T prime, this heterozygous version, this is tabby. And what it is it's not mixture. It's not a combination of tan and black, kind of a brownish color. It's patches of tan and black, equally brilliant. And in chickens, you can kind of sometimes see white and black feathers in a checkered pattern. And the way we would do this is if you had a tabby cat crossed with another tabby cat, what might the offspring be? And so we can do this genetically with this T prime, that's tabby, crossed with another T prime. And we do the Punnett squares the same way that we've been doing them in all the other versions, here and here. So there's the tan allele, the black allele, the tan allele, and the black allele. And we get this kind of classic combination that we've seen every time we cross heterozygotes. So this one here would be black, that's tan. This cat here would be black. And these ones here, there would be a 50% chance that they would produce a tabby cat. So that's the other kind of version that happens with codominance that you're going to see in this unit. All right. Happy studying. As always, let me know if you have any questions, and good luck.